Hi, I'm Sarah.
I'm Bowie.
I'm Blake.
I'm Kyle, and this is our mathematical model for culture of human neural stem cells to treat neurodegenerative diseases.
Neurodegenerative diseases like Parkinson's, Huntington's, and Alzheimer's debilitate millions worldwide, with Alzheimer's impacting 6.5 million people in the U.S. alone, making up 60 to 80% of all forms of dementia. These diseases have critical economic and societal implications, as there is currently no known surgical or pharmaceutical cure. Furthermore, without an ample supply of validated stem cell therapies, large-scale solutions remain quite limited and costly. These diseases are characterized by the progressive degeneration of neurons over time, including loss of structure, function, or the death of the neuron entirely. Consequently, scientists have explored using stem cell therapy as a means of minimizing the disease's influence on a patient's functionality. One such category of stem cells used for therapy include neural stem cells. Human neural stem cells are multipotent adult stem cells, which generate the main phenotype of the nervous system, allowing them to differentiate into neurons, astrocytes, and oligodendrocytes. Neural stem cells are also self-renewing through neurogenesis, which allows these cells to respond quickly to injury or damage caused by Alzheimer's, making it easy to replace lost or injured cells. Therefore, understanding how neurostem cells can form new neurons in patients with Alzheimer's is of our utmost interest and importance. This led us to our main objective, in which we strive to develop a mathematical model to track the growth and collection of neural stem cells in both a series of batch flasks and a bioreactor. For each system, we tracked inputs like glucose substrate, oxygen, and ammonia, while also considering the costs each system would require to bring to complete functionality. For our first model's small time periods and small amount of target cells, the batch flask system was superior to the bioreactor in terms of the yield and overall cost. However, given the substantial number of cells required to treat one person, roughly 6 million cells per kilogram, it meant that our produced number of cells was not nearly enough to support cell therapy with neural stem cells. Thus, we modified our batch flask system to a flask expansion system, splitting each flask into 2 every 12 days. Using this new model, we determined that the stirred tank bioreactor still produces our needed number of cells in significantly less time, about 63 days compared to about 90 days. In the case of the bioreactor, however, it used significantly more media because of the need for constant input, ultimately leading to a cost almost 6 times higher than the batch flask system, despite using 127 total flasks.
The basis on which we derived the equations for our model is the biomass equation shown here. The empirical formula for cellular biomass, CH 1.743, O 0.316, N 0.146, was determined using the average composition of a dry mammalian cell. On average, the composition of a mammalian cell is 5% nucleic acids, DNA and RNA, 60% proteins, and 13% lipids. A nucleotide, the monomeric component of nucleic acids, has an empirical formula P1N5O7C10H14. Assuming a guanosine and monophosphate base. The average amino acid has the empirical formula N1O2C5H8. The average lipid has an empirical formula C18O2H36. Finding the weighted average for the contents of the cell and normalizing it to carbon, we determined the empirical formula for cellular biomass. Once we found the empirical formula, we used the biomass and toxin yields, as well as a respiratory quotient of 0.97. We balanced the equation while normalizing all the values to have 1 mole of glucose. Here are our schematics for the 2 different systems we will be modeling, with the batch flask model on the left and the bioreactor model on the right. The batch flask is a closed system, and thus there is neither input nor output of any substances involved in the production of biomass. Rather, there is initial concentration of each necessary reactant, as well as cells, which are either depleted or increased over time. The biomass equation serves as the consumption term for our extensive property of interest, molecules of cells, while consumption can be modeled by a literature-derived cell death rate, KD. Likewise, the biomass equation also models generation of carbon dioxide, water, and our toxin lactate, and consumption of ammonia, oxygen, and our substrate, glucose. In our bioreactor model, there is a constant input of both glucose and ammonia, while oxygen is sparged in at the same rate. Additionally, there is an output flow of substrate, toxin, carbon dioxide, ammonia, and cells to be collected downstream. The generation and consumption terms are the same as in the flask. When modeling the cell change within the flask, we made use of Monod equations to determine the growth of cells. Modifying a literature value for the maximum growth rate of mammalian cells, we are able to take into account the effect due to reactant consumption of substrate and oxygen, as well as the toxin limiting effect as it is produced. Lastly, we factored in the cell death rate due to metabolism mentioned earlier. We use similar logic for each of the reactants and products of our system, especially our chosen toxin and substrate. We chose to model this using the biomass equation, which consumes glucose and produces toxin at a proportional rate based on the yield coefficient to the production of cells. Because we use the substrate as the base coefficient in our balanced chemical reaction, the numerator for glucose is 1. In each other case, we divided the coefficient of the term of interest by the yield coefficient. For O2, CO2, and ammonia, we use the same concepts as before to apply to our yield equations, referring to the ratio of yield coefficients for each element as n. When assembling the equations for the bioreactor, we chose to take those used in the flask and simply modify them based on the added input and output of the system. Here, we added an output term for the cells from the bioreactor, which was calculated by multiplying the ratio of molecules of X per volume of the bioreactor by the volumetric flow rate. Likewise, we created a new term that represents the collection of cells once they are removed from the system. In order to determine the volumetric flow rate both in and out of the system, we calculated it by multiplying the volume of our system, V, by the dilution fraction D. In order to minimize costs and maximize output of cells, we have performed a parameter sweep for several different values of D. Though lower values such as 0.1 maximized our yield, the time taken to reach the maximum decreased as we increased the dilution rate. However, once we reached a value of 0.3, the materials were replaced before they could be used. This would waste a ton of resources and increase the price, as well as decrease the yield, so we decided to use a dilution fraction of 0.25. Just as we did for cells, we modified our original product equations to include a similar ratio of output based on our calculated volumetric flow rate. For our reactants, we did the same thing with one exception. Because these products are either flowed or sparged in over the course of our experiment, we must include an input term. Using the same volumetric flow that was calculated for flow out, we multiplied each by the concentration of the compound in question that we would be pumping in.
In both our batch flask and chemostat systems, we wanted to determine which of our reactants between glucose and oxygen was the limiting reagent. We decided to graph the substrate and oxygen concentrations over time, leading us to the conclusion that oxygen is the limiting reagent. The glucose concentration in the flask decreases exponentially to a steady-state value of 3.5 micromolar, while the glucose concentration in the bioreactor is relatively constant around 0.125 molar for the duration of the reaction process. This indicates to us that glucose may be a limiting reagent of the batch flask system, but not necessarily for the bioreactor. In the flask, oxygen concentration decreases logistically from 0.0025 molar to 0. Biologically, this represents the rapid use of oxygen by growing cells compared with the incredibly low diffusion rate of atmospheric oxygen into water. In the bioreactor, the oxygen concentration initially increases to a maximum concentration of around 0.02 molar and then decreases to a steady-state value of 0.078 millimolar. The low steady-state values indicate that oxygen is likely a limiting reagent in both systems. Because our system is at steady state in the bioreactor, the accumulation of oxygen is 0. Using the mass balance accounting equation for oxygen for the bioreactor, we can see that oxygen is being consumed as quickly as it is inputted into the system. Using a differential equation solver in Python, we graph the number of cells in each of our two models until they reach steady state. As can be seen in the graphs here, the batch flask reaches its steady state a little less than 2 days earlier than the chemostat, but the amount of cells produced in the chemostat were around 1 order of magnitude greater than that of the batch flask. However, when the flask reaches steady state, we know that it has run out of the reactants needed to produce more cells, and that the number of molecules of cells in the batch flask has reached the maximum number that can be gathered from the experiment. On the other hand, the steady state for the chemostat combined with the constant input of cells signifies that only accumulation has stopped increasing. The generation of cells due to the constant input of glucose, oxygen, and ammonia is offset by the output of reactants and produced toxins in cells. It is clear that in the long run, the bioreactor is significantly more successful in terms of the collection of cells. However, we were curious to examine what happens in the initial period of each system running. It can be seen here that in the first 10 days, the flask actually outperforms the bioreactor in terms of produced cells, despite the fact that each are initially seeded with 10,000 cells. This can be explained by the fact that the reactants are flowed too quickly from the chemostat for the reaction to produce a significant amount of cells to be collected. However, this effect is not permanent. Around the 10th day of the experiment, the flask reaches its carrying capacity and plateaus, while the rate of cell collection for the bioreactor increases significantly. At around 10.5 days, the number of cells produced by the bioreactor exceeds the number of cells produced in the flask for our model.
The main goal of our model was to determine the feasibility of culturing human neural stem cells for cell therapy in the treatment of Alzheimer's disease. To do this, we investigated the time, reagents, and cost needed to treat one person. Current clinical trials in the treatment of neurodegenerative diseases and traumatic brain injury cite 6 million cells per kilogram as an effective treatment for these disorders. So we chose to look at a 120-pound human, which translates to about 50 kilograms. And for this, we would need about 320 million cells. Here we have plots of our collected cells from our chemostat model and from our batch flask expansion model. On the left is the chemostat, which after steady state exhibits linear growth in collected cells. And on the right is our batch flask expansion, where every 14 days, one flask is split into 2 new flasks, and the cells from the original flask are collected. The red dot represents the 320 million cells we would need to treat one person. Immediately, we can see that the chemostat model takes significantly less time to reach this 320 million cell count. So, looking at the time, the chemostat would only take 63.1 days to reach this count, while the batch flask expansion takes 89.6 days. This is about a 30% increase in amount of time required to collect these cells, which is a significant amount of time when treating a disease. However, we should also assess the reagents used and the cost associated with the model to determine which is the most feasible. So, although the chemostat model took less time, it cost significantly more and used much more media. The most significant increase in cost for the stirred tank bioreactor came from the amount of media used, which was due to the constant inflow of new substrate to allow the cells to continue to grow. This model looks at only the time and cost to treat one individual. However, there are as many as 6 million individuals living with Alzheimer's disease in the United States alone. Using the time and cost for our one-person model, we can make a crude estimate for the amount of time and money that would have to go into providing this cell therapy for an entire population, which would take many years and cost many billions of dollars, and would probably be unfeasible. In conclusion, the optimal method for culturing neural stem cells is largely dependent on the goals and purposes for culturing. For short times and smaller amounts of cells, a flask method is optimal, and it is much more cost-efficient due to the smaller amount of media used. However, if more cells are needed, it is better to use a bioreactor. However, for the required amount of cells to treat a neurodegenerative disease such as Alzheimer's, for use in cell therapy to treat neurodegenerative diseases such as Alzheimer's, neither of these methods would likely be able to produce enough cells to treat an entire population. A better strategy might be to explore a system such as the Corning Hyperflask, which combines the high yield of a bioreactor with the low media demand of the batch flask system. All in all, the culture of neural stem cells may not be a viable option for the treatment of neurodegenerative diseases. If we do wish to use neural stem cells in cell therapy, we must demonstrate that they are both effective and necessary, and that these factors outweigh the cost and time it would take to culture them for an entire population.